the University of Oxford, and there are two professors of mathematical biology at Oxford. So one is in the mathematics department. It's Philip Maney. He's one of the co-organizers of this meeting. So unfortunately, he couldn't be present. The other one is in zoology, and has been done traditionally for many, many years. So in the case so many of you might know what Philip Maney does, because he does pattern formation. He's very well known for that. In the case of Mike, so he's an expert on population dynamics. And he's an excellent public speaker. And he was chosen precisely because we wanted to hear from somebody who actually does mathematical biology in a biology department, and at the same time is an effective public speaker.
Okay, great. Thank you very much. Thanks very much for that introduction. Thanks very much for the invitation. It's a great pleasure to be here this morning. As I said, I'm based in Oxford. I'm based in the zoology department in Oxford, and in my group we do mathematical biology. I grew up as an ecologist, so one of those theoretical ecologists that might have been rejecting your papers before. I'm sorry about that, but that's what I do. So I'm going to motivate my talk with lots of different examples of sorts of stuff that we do in my group and the sorts of research that we get onto. And the title of my talk is Scaling the Heights of Biology with Mathematics. So I'm going to be thinking about how we move between different, in like the last talk, we're emphasizing the macro from the micro to the macro. And it's really thinking about that and how we actually use our mathematics to be able to help us do that. So what am I not talking about is this. Classically in biology, whenever you mention the word scaling to anybody, it always puts in their heads about this idea of allometry. So this idea, we've got fiddler crabs, and there's something to do with their body size and how big their pincer is, and it's always thinking about how can we think about that? How do we sort of plot out the relationship between a body size and a log scale and a pincer on the y-axis, and see whether there are differences, and as body size grows, does pincer size grow in a different way? And that's not what I'm talking about. I'm not going to be talking about this classic ideas in allometry. Not that it's anything wrong with it, but actually it's just static, and I think it's a little bit dull. But I'm not going to. So we're just going to move away from thinking about that allometry ideas and really move into thinking about dynamics in some way. So what you're going to expect this morning from me is that I'm going to sort of show you how we can do slow-fast, what slow-fast approximations are. I'm sure you're all familiar with them, why they're useful, and why they're not useful as well. So I'm going to motivate that with an ecological example. I'm going to also introduce you to some functions, also scaling through functions. Again, maybe not rocket science for everybody, but I'm going to motivate that with an idea about the blood stem cell system, and some work that we've been doing on that area, and really thinking about how we can understand links between different aspects of the hemopoietic stem cell system. And I'm going to introduce you to sort of like predator-prey interactions. This is sort of like bread and butter for me, so I might, if I get dull, but I won't. I will get into this, and we're going to motivate this with a little bit of audience participation, so be prepared for when we get to that. I'll need your help. As we get through a little bit further into this, I've got more approximations, so some clinical stuff that we've been involved with by modeling mood and bipolar disorder, and then moving a little bit beyond biology into the realms of biocontrol, and when I get to that, I will get there. I'll explain that. So slow-fast dynamics, why are they really important? So why do we need to be able to understand these? So I'm going to motivate that with a particular ecological example. This is one that's very close to my heart. It involves a particular moth, cinnabar moth, pretty moth. It's out in the middle of June. It's a specialist herbivore. It's caterpillars. I'll show you some pictures of those in a moment. It feeds on this plant, which is ragwort. And ragwort is particularly nasty if you're a horse. It causes blisters in the mouth, but it's full of very nasty chemicals that the moth caterpillars are able to sequester, and they have this black and yellow aposematic coloration, so it stops them being eaten by things or birds and makes birds sick. But we were interested in the dynamical interaction between this herbivore and this plant, essentially. And this is a math bio session, and I am not going to shy away from putting up equations, and I'm not going to shy away from any math. So we're just going to sort of walk you through this. On the left-hand side, I have two ordinary differential equations that explain sort of generically and maybe quite simplistically this interaction between a herbivore and its resource. So big R is the resource. It's going to flow in. It's going to change through time, gradually through time, infinitesimally small changes through time. We've got it flowing in at some rate lambda. This resource is just going to flow into the system. I've got some consumption by herbivores on the resource at rate alpha. That's just how fast they're eating the stuff. And if you don't eat it and it stays around too long, it's just going to die. And as everything is in biology, in mathematical biology, it's going to decay away exponentially. And we'll get more into that in a moment. So we have this sort of coupling between what the herbivores eat and how it impacts their growth rate. So this is a positive on the right-hand side of this. So that means the population is going to grow if alpha is positive. And again, if they don't do anything else, they're just going to die. And that's what's going to happen to this. So we're very simple plant-herbivore interaction. So we might think, actually, the mathematics behind that can be quite complicated. We may not, but if we want to get a handle on that sort of dynamic, things may be operating on different timescales. The resources may be flowing in really fast, and the herbivore dynamics may be operating on a different temporal scale. And that's the problem in biology, and that sort of underpins everything I talk about, is the difference between timescales, the separation of these sorts of timescales. So we've thought about thinking, one way to approach this is actually to separate out the slow and fast dynamics. So we can do that. We can think, actually, if we think that the resources are turning over really fast, then actually the herbivores may only ever see an average of that. And one way to approach that is with this simple approximation of the slow-fast dynamics. It says if resource consumption exceeds the loss rate, so what's been eaten is greater than what's not been eaten, then we can simplify this dynamic out. We can set this, so is the fast dynamics to some sort of equilibrium, and we can solve that for some average rate of resources in the system. Well, that's pretty cool. It tells me it's a ratio between the growth rate and the consumption rate. Not rocket science, but it's pretty interesting. And we can go out and we can look at that. If we knew these sorts of things, we know how many herbivores are out there. We can work out the flow rate of resource into the system and how fast these herbivores are working. And we may be able to get some idea of some equilibrium level of herbivores. Well, we can do a little bit more. We can do a little bit more fancy maths, and it's fancy maths only up to a sort of like school sort of standard. So I can take my herbivore equation, and now instead of having the resource consumption rate, I can approximate it with, so instead of having R in that as a dynamic in that, which is changing through time, I can take it at its equilibrium level. So what I've done is to replace R with this term in the middle, the ratio between the growth rates and the consumption rates, and it turns out to simplify the math. So I can cancel alpha and G on the right-hand side of that. So I'm left with this little expression, which is getting simpler all the time, and I can solve that. So what's on the right-hand side, and this is particularly aimed at school kids because we do a lot of this, is actually when I was at school, my maths teacher continually told me, so Mike, you need to, or no, actually she wasn't that polite. She said, Bonsall, you need to show your working. So there's my working for getting from that equation to this equation. So what does that tell us? Well, actually, we need to be able to sort of graph that thing, or we need to be able to be clever enough to actually approximate what this function, or to be able to see what this function looks like. So I'm sure, in a room of mathematicians, I don't need to do this too hard, but actually we've got looking at G of T, what the sort of dynamics of the herbivore are through time, essentially. So I've got time on this, on the x-axis, number of herbivores on the y-axis, and it will plateau out. And it will plateau out at some ratio between the flow rate of resource and the death rate of the herbivores. So I end up with some equilibrium. I end up with actually this, what we should expect with a slow-fast dynamic is that the herbivores should grow, this moth should grow to some nice steady state. Well, that's pretty cool. So if that's the way the system's working, that's what we should see. One of the very cool things, and one of the things we like to do a hell of a lot, is actually to combine our thinking, our mathematical thinking, with biological realism, or biological data. So this is a study that was started in the 1970s and ran up until the into the mid part of the last decade. And we've got the log numbers of this moth and the plant on the y-axis through time. The black line is the moth dynamics, the dotted red line are the plant dynamics, and these are the caterpillars, as I was talking about. You can see they're really voracious. They will strip the ragworts of all their flowers and leaves and just leave little stems, essentially, and then they'll wander off and find others, essentially. And they're sequestering all of this nasty stuff that's in these plants. But this is their dynamic. This is the dynamics that are recorded, and we have data, we have time series data from other places. One in the UK looks very similar. The moths and the herbivores show these sort of long period cycles. So then we have a problem, because I've just told you that these slow, fast dynamics, and we know that the dynamics of the plant can operate on a lot faster timescale than the moth. So our prediction from our equation would tell us that actually the black line, the moth dynamics, should be at some steady state. But they're not, and that's a problem. And it's a problem for two reasons. One is that actually we do a lot of this slow, fast dynamic stuff in mathematical biology. We assume we can average over those fast dynamics and just look at the slow dynamics, and that's a real problem because actually we lose a huge amount of information. We lose the variance in the fluctuations in this dynamical system. So actually this approximation, while it's good, isn't sort of really that good. And I'm going to come back to this as we get through the talk. I'll sort of illustrate why we might do that. So one of the things we can then do is actually think, well, actually, what is the actual data telling us? So like these inverse problems that we've talked that have been mentioned in the last couple of talks have illustrated how we can take data and make inferences on the underlying dynamical patterns. So one of the things is that we make a very simple assumption that actually the numbers of herbivores or the number of plants are only in those simple ordinary differential equations are only dependent on what was around in one previous time step. But we can sort of like mathematical biology isn't just about mathematics. It's also about statistics, and we can blend all those together into thinking about how we can blend our thinking about maths and data together. And we can do that. We can analyze this sort of time series. We can take lots of different ways in which we can approach that time series statistically. So we're taking a model fit. We sort of say, how good is our model at describing the data that we see? And we can do that both for the moths and for the plants. And when we do that, when we do sort of go through these ideas of selecting the best sort of models, what we find out is that the dynamics of the system are a whole lot more complicated than we might have thought about. So we've got basically the moths. Actually, yes, the moths are dependent on how many were around in the previous time step. But there's also some complexity. There's a little bit more detail in that. We've also got them dependent on the plants. Well, that's good because they eat the plants, so it's good that that falls out of our model. But it's also more detailed than that because they're not actually dependent on the plants that are around now or in the last time step. They're actually dependent on the plants that were around 2 time steps ago. So there's some long lag in that. And then there's some even more interesting biology that comes into this. There's actually the moth dynamics, the moth numbers that are around 2 time steps ago actually factor into driving up the patterns in the changes in the black line in the moths. And then we've got the sort of plant dynamics, and the plant dynamics, so similar sort of, so we've got actually the numbers that were around last year actually is important, or in this years that are important, but also the numbers that are around 2 time steps ago, and actually the moth densities as well. So we now understand that this is actually quite an intricate, coupled system where that simple approximation may not necessarily work. And we'll get into how we can solve that in this next section. So I'm going to talk about functions. I'm going to talk about how we can use functions to scale between different levels of organization in biology. And as I said, I'm going to motivate this with some work on the blood system. The hemopoietic stem cell system is fantastically complicated. We should just pay homage to how great our blood systems are because they do everything for us, do everything that keeps me breathing while I'm talking to you, just about, and helps me sort of fight off infections because it's all producing all of those sorts of immune cells. So we have in our bone marrow pluripotent stem cells that sort of will differentiate out into a whole set of progenitors. So we have lymphoid progenitors that go on to form our immune systems, and myeloid progenitors that go on to form the complexities of our peripheral blood, so the red blood cells and the platelets and the macrocytes and all this sort of stuff. As we move down that, we get sort of increasing levels of differentiation. But there's the sort of very cool dynamical understanding of this is what is the sort of like demand and how is that controlled? How does peripheral blood or your peripheral immune system tell your bone marrow actually, hey, we need more blood cells, we need more red blood cells, or we need more white blood cells. We're under threat here. We need you to sort of ramp up the activity. That's a problem. That's obviously a biological problem, but it's also a really interesting mathematical problem. So the transitions, we can think about this sort of like stochastically or deterministically. We can sort of set up sort of equations and models to sort of describe this. And classically, the classic piece of work was done about 50 years ago, or maybe 60 years ago actually, in the early 60s by James Till, who demonstrated that actually this all kickstart the sort of stem cell cottage industry in stems and thinking about stem cell biology. And he was interested in thinking about that there were stem cells that would differentiate and there were stem cells that would just sit there in the spleen. And he showed that actually he did some really cool math bio in the early 60s to fit different functions, different stochastic functions to these sorts of patterns he was showing. And illustrated that actually there are stem cells in the system that are just quiescent. They just sit there, they're not really doing anything, they're just really quietly hanging out, and when the system gets ramped up, they kick in, essentially. So that sort of lays out some really interesting sort of ways of thinking about this. He sort of like, just thought about this quite simply, but obviously we can think of a whole set of different transitions that can go on. And this is my schematic. These sort of bluey, grey, greeny things are stem cells, and the reddy, blacky things are progenitor cells, so they've some differentiation. The yellowy things are differentiated cells. So we can think of there's a demand in the system is saying, actually, we need more stem cells, we need more stem cells. So it goes, actually, we go from 1 stem cell to 2 stem cells. Yeah? Or we can say, actually, what we need actually is you to make actually some progenitor cells. So a stem cell sort of stays the same and it produces a new progenitor cell. But then there's a whole set of other transitions that we can think about. We can have, they might be really strong. We might be saying, actually, I'm under attack. I've been cut. I need more red blood cells. So the blood system kicks in and says, look, mate, go from 1 stem cell to 2 progenitor cells and onwards down to those red blood cells. They might die. Well, that's a bit sad, but they might die. Progenitor cells, there's always some, may actually proliferate a little further, they may pass away, and those progenitor cells may differentiate into more specialized cells, more T cells or B cells, platelets, erythrocytes, whatever. There's more specialization, and they will decay away. So there's this whole set of different transitions that we might want to take into account. And there's even more interesting stuff that goes on. The background that the bone marrow sits in is actually really important and will govern the strength of these signals. So this is just another schematic to show sorts of positive and negative feedbacks that can operate in this system. So we've got basically things that actually there might be a negative feedback saying, hey, we're okay, we don't need anything, which sort of down regulates the stem cell activity. There may be actually sort of a kick from down here that says actually the fully differentiated cells are rather depleted and they need more. And there's a couple of functions here, and then I'll get to the maths, where we've got sort of like these two functions that are phi, which has got P, D, and B in it, so the progenitor cells, the differentiated cells, and the background cells, all influencing the strength and the magnitude of these sorts of feedbacks. And the difference between the sub 2 and the sub 3 means, am I producing a simple response where I get this response, where this is for one of those feedbacks, where it's a slower or sort of more weaker response, where a stem cell is still maintaining itself and just making one new differentiated progenitor cell, or there's a more stronger signal, say, hey, I need 2 more, I need a lot more progenitor cells coming out of this. So we can capture that. So that's the schematic about all of these things, all of those different processes I've just talked about. And here, for just proof of existence, there's some equations that show that, essentially. So we've got actually the stem cell, progenitor, and differentiated cells. There's a whole set of interesting maths there to do with the fact that they live in niches, the fact that these functions, phi 3 and phi 2, are describing the strength of the demand for the stem cells to do something. There's some deaths on either side of that, and we just work our way balancing those things out between the progenitor cells and the differentiated cells. The really interesting thing is about how do we actually describe these functions, these 5 functions. So there's lots of ways we can think about that. So we can think about, if we go down to peripheral blood, we can think about, well, what's the number, what's the density, proportion, whatever you like to think about it in terms of the lymphoid cells that are operating in the system. And then should they be we think about this in homeostatic environments, how should they be balanced out in let's put it, in a healthy patient? So we think so that might be sort of like the number of myeloid cells that are made in proportion to the lymphoid cells could just be independent. They may just be doing doing completely separate things and not actually talking to each other. But then it could be proportional to need. It could be actually linearly proportional to need. So actually, if there's more lymphoid cells, you need more myeloid cells, or it might be non-linearly proportional to need. But we just don't know. We don't know what sort of patterns might come out of that. So again, we can go back to our maths. And we start by sort of formulating a function. So this is a functional response. If you're an ecologist, you'll know what those are. If you're a mathematical biologist, you might know them. But they're just relating something to do with the system. So that in this case, the proportion of myeloid cells with respect to other things that go on in the system. In this case, the proportion or number of lymphoid cells. So I have some mathematical formulation to do with that. So some ratio, kappa is the relationship between the myeloid and lymphoid cells at homeostasis. And then there's some sort of some idea, we want to be able to capture this idea. There could be potential for nonlinearities in the system, so we're going to raise that to gamma. And there's some control parameter alpha on that. So rho is the sort of like the proportion of myeloid cells that we're going to get out of that. So we can solve that. We can sort of do some, not even fancy maths, we can do some algebra on that to show out that actually there's a set of, so if we know this ratio and we know what the homeostatic level is of these things, and we can work out what this parameter could be or should be. What it shows is that there's a family of parameters that are consistent with homeostasis. There isn't one set of parameters that are going to, so I can have different values of alpha and gamma that all give rise to a homeostatic environment. So that's pretty cool. So I can choose different values of gamma and different values of alpha, and the system, this homeostatic, the homeostasis in the blood system will be very nice and steady state. But what does that imply for these particular functions? So as I said, we've got this particular formulation. It's just the way that we've thought about this. What does it mean for the sort of formulation between the proportion of myeloid cells, or the number of myeloid cells, and this ratio, or the numbers of myeloid versus lymphoid cells? So for homeostasis, we can end up with a whole set. Even in this homeostatic environment, we can have a whole set of different demand control functions that drive our hemopoietic system, depending on sort of like the parameterization of this, which is pretty cool. So we can go from everything that was independent, so the response for myeloid cells could be independent of what's already around, or it could be non-linearly proportional to need. So that sort of puts us into a really interesting situation, because actually the dynamics of, and I've used this word a lot now, so we've used complicated and complex, which is pretty interesting in thinking about actually, even in homeostasis where we think everything is in steady state, so at the macro scale everything seems to be all at some steady state. Actually what's underlying that is a whole set of dynamics, and being able to approximate those and understand those with these sorts of functions is pretty cool. So I'm going to get a little bit more into this idea of this function and this functional response. And again, it's a classic way where this was actually, for a theoretical ecologist, came about, was thinking about predator-prey interactions. So how do we relate predator behavior to predator dynamics? So I'm linking that, for that sort of like going back to that sort of resource consumer system I had at the start, where you've got herbivores eating plants and affecting their growth rate. So we've got a whole set of predator-prey interactions that exist in the natural world, and they all depend on some sort of behavior. They all depend on us the predator being able to find its prey, do something with that prey, and convert that prey into new predators, essentially. But those things operate on different time scales, and it's really being able to think about how those sorts of time scales play out. Classic Lotka-Volterra type dynamics. I probably don't introduce this to anybody, but just in case there's anybody in the room who may not. dm by dt, the changes in prey. dp by dt, changes in predator with respect to time. So I've got prey. In the absence of anything else on the right-hand side, prey are just going to grow exponentially. And I'll show you what that looks like very shortly, just in case. The only thing that happens to prey is that they're eaten again by predators at some rate alpha. There's some conversion rate of eaten prey into new predators at rate c, and predators just die. Classic Lotka-Volterra type dynamics. So in the absence of prey, our bunnies will increase exponentially, and we'll be knee-deep in bunnies. And there's nothing in this simple system. Obviously, that doesn't happen, but that's not the point. What I'm trying to illustrate this morning is that we're thinking about this link between predator behavior and predator dynamics. So then the natural question is, well, what happens? What happens as a fox enters a system and bunnies start getting eaten, and sort of like this behavioral response of the predator to prey, and then there's a numerical response that the population size of the predators either shrinks or grows. And how do we understand this link between these predators' behavior and these prey, and the density of prey? So the way we're going to do that is with an exercise. And as I said, this is where we need audience participation. So the way I normally do this is with chocolate, but on my travels, I couldn't actually find chocolate. So I did manage to find, so I normally motivate my talks on Math Bio with lots of chocolate. But I did find M&Ms, crispy M&Ms, which are almost as good, essentially. So I need 6 volunteers. And if you don't choose, I'll just come and pick on you. So you might as well, if you want to eat crispy M&Ms, and as soon as somebody puts their hand up, the bigger number of M&Ms they're going to get, you're going to get. Are you up for a volunteer? Yes. Okay, take a seat. Great. I'll come to you. Don't worry. You don't have to come to me. I know audience participation is a little bit scary when you have this. So you can have this bag. It has 75 crispy M&Ms in it. Okay. Thank you. Okay. Just hold on to that. Right. So now we're going to go down. I have 20. Okay. You're going to get 20. M&Ms. So don't take them out of the bag, because I know with these ones, they're a little bit more rolly round than the... Sorry, I think... Oh, no. I think that one's got 20 in it. Right, so we're now down to 10. Who would like 10? Toby, you don't seem to be doing much. There you go. Have 10. All right. Right, have I got 2 more? 5? Anybody want to do 5? Oh. Yeah, excellent. Thank you. And I have 2. You'll take 2. Excellent. Sorry. Thank you very much. Now you'll need to be close to somebody else. Like if you don't know your neighbour, now is a great time to introduce yourself to them because you're going to need their help. All right? So you need to find somebody to sit next to him. All right, because you need that. So the idea is that in your bags of M&Ms, you just open them. I normally do this on paper, but the room's not really set up for doing this. But actually, in your bags, you have different numbers of M&Ms, these little chocolates, little crispy chocolates. Okay, proviso, health and safety. Actually, if anybody has, these things are made in factories that have nuts and peanuts. So if you have those sorts of allergies, maybe pass them on. Please don't eat. I don't want anybody to have sort of like an incident, a sort of anaphylactic shock on me, because that'll just be awful. All right, so the idea now is those are your prey, and the mathematical biologists are predators. All right, and we're going to see how many you can find in a finite period of time. All right, so the idea is that you're going to close your eye, you're going to open the bag. I can illustrate with my bag. My bag's empty. You're going to open your bag, hold it in, give it to the person sat next to you, just to hold. You're going to take your index finger. This is your index finger. This is not. As Toby pointed out, we have digits. We have 5 of them on one hand, 5 on the other. One of them is your index finger, right? Take your index finger, tap it around the bottom of the bag. Okay, that was going to be easy, but when you find an M&M, use the opposable thumb to pick it up and give it to your partner, the person next to you. Okay? With your eyes closed. It's much more fun if your eyes closed. Even with the small kids, we do this. We blindfold them, and they think it's fantastic, right? So the idea is you're going to see how many you can find. You're going to take your index finger, you're going to tap in here. When you find one, you're going to lift it out, give it to your partner. You need to remember how many you're in your bag to start with. I told you a number as we went around. I want to know how many you get out at the end, because we're going to do some real-time science here, essentially. We're going to do some mathematical, I'm going to do some statistical analysis to see how good a predator mathematical biologists actually are. Okay? So I'm going to type. So just again, index finger in the bag, tap, 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 find an M&M, lift it out, give it to the person next to you, finger back in the bag, tap, 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 find it again. Okay? Yeah? Yeah, okay. We're all practiced. You all ready? Eyes closed. Okay. I've got a timer up here. You ready? Everybody happy? Sorry, I can't hear. Everybody? Yeah, just give it to the same person. We can share them round afterwards if you want to have M&Ms. We can certainly feed people. I know I stand between you and lunch, but actually these things they're probably quite nice. Okay, you ready? Right. So right. We're all going to do this together in a timed fashion, essentially. So if you've already taken M&Ms out of your bag, put them back in the bag, and we're going to see how many we can find. Ready? Yeah, I'm going to give you a shout to start and a shout to stop. Okay? Everybody ready? Yep. Close your eyes. Let's go. Yeah, okay, if you find them all, you're done, basically. Yeah. You can eat them once you're done, basically, as long as you know how many you've got. I need to just know the number that you found. Yes. It's like being at school, isn't it? I did this one time, I had pennies, and so the girls in the class said, "Sir, can we keep the pennies?" Like I'd given them 150 pennies. It's like, yeah, sure, I don't want to carry those home. Okay, let's stop. All right, how many have you found? You found 2? Who had 5 in their bag? Did you find them all? How many did you find? You have 5? Excellent. Who had 10? How many did you find? 10. You find all 10? Excellent. Right. Somebody had 20? 14. Excellent. And somebody had 75, who's going to be very sick on how many have you got? Those are the ones you found. How many have you got? Oh, okay. That was the idea, is to count them as you went along. I know it's, yeah. All right. So I have, it's hidden, but you can't see it, but I am using a little bit of R code just to do some model fit on this, basically. So while they're counting, I'll show you what a made-up number for the last looks like, essentially. Oh, it's not quite good enough. But anyway, so up to this, we'll fill that number in at the end, basically. 36, excellent. That was good because I put 32 in there. So with a little bit of biological error in there, we're okay, essentially. So you feel free to eat those. That's perfectly fine. So we had the number of M&Ms on this axis. Sorry, this is fallen off here. So I have the number that were in the bag. And this is the number that you caught, basically. And the idea is then thinking about this functional response. So the blue dots are the numbers that you caught, the numbers that you found, and this red line is the fit, a nonlinear fit, using this, being able to take a whole range of different sort of functional forms and just finding out what's the best fit between the data and the model. And it turns out to always have this particular shape. Right? It always plateaus out. I do this, as you get more prey in the system, there's only becomes a finite number that can ever get caught. So that's why I put a lot in the bag. If somebody will want to ask me, why do you choose these sorts of numbers, Mike? Well, actually, it turns out that's how many are in the M&M bag when I buy it, essentially. So they actually can use them all. But actually, as I keep going, if I gave you 100, or if I gave you a bag of whatever it is, 130 M&Ms, you'd still only find about 30 of them in that sort of finite time of about 45 seconds. So that introduces to this idea that actually predators are limited. The idea is that actually predators cannot continually search for prey. They have to stop, they have to do something, you have to convert it into new predators, and it has this particular relationship. Well, this particular relationship, so if you twist it around in your head, rather than having the numbers on this axis, I have the proportion of prey eaten, then this looks very different. This looks very different. So actually, down here, when I have this, where everybody finds all their prey, obviously the proportion is 1, but that decreases as prey density increases. So I end up with this negative relationship. And we know from theoretical ecology, lots of work since the 1920s, that sort of relationship, where you have positive feedback or inverse relationships, is going to destabilize dynamics. So it's going to move our system from something that's unstable to something that's something that's stable to something that's pretty unstable. Let me just go back to my little talk. Okay. So when we go back to our moth system now, we have a much more robust understanding of why we see these sorts of dynamics. That Lotka-Volterra model tells us when we have this type of function response between the behaviors of the predator and the dynamics, we expect to see cycles, and we've got log abundance through time, and we can see cycles. Obviously, there's still nuances of the system that we haven't quite captured with that model, but we've captured the general pattern. The general pattern that actually there are long cycles because of this scaling between behavior, something that's operating at a different temporal scale than the dynamics. I think it's pretty nice to be able to do that. So again, I want to move into just another bit of area where we can use approximations. And this, I think, is a bit... obviously, I think all my work's cool, but if there's any other bit of cool work, this is it. Okay, so I have been working with a very cool colleague who is based here in Stockholm at the Karolinska Institute for about 10 years now. I've known her for a very long time before that, but we only got working about 10 years ago. And Emily is a clinical psychologist, and she is interested in a whole range of sort of mental illnesses that present themselves, everything from sort of bipolar disorder through to PTSD. And she said she's a clinical psychologist. She worked in a psychiatry department, and we got interested in doing this, what has become quite a vogue thing at the moment called computational psychiatry. But we're interested in actually being able to apply mathematics to be able to understand the sort of the dynamics of mental illnesses, and we've worked on a range of different things. But the one I'm going to present you today involves patients who have bipolar disorder. And one of the very cool things, Emily was originally based in Oxford. In Oxford, the psychiatry department, the psychiatry group in Oxford have an online outpatient sort of data collection process called True Colours. And bipolar patients are asked to report their mood. That might seem a strange thing, but actually it's a very well proven clinical instrument for diagnosing risk of episodes in bipolar. So they have these very long time series of a range of different clinical scoring systems, and this one's called QIDS, Quick Inventory for Depressive Symptomologies. It's a 16-based score system. It's a multiple choice thing, and you can sort of take that data in lots of different ways, and we can aggregate it up. And clinicians use it for looking at the profile of bipolar patients and seeing whether they're at risk of episodes of depression or mania. But we were just interested in just applying some very simple mathematical thinking to this. In the first instance, the first piece of work we did was really a statistical exercise in just can we describe patients with using these time series? Are there different structures in the time series that we see between patients who are high risk or low risk of bipolar when they've been classified by a psychiatrist? This is a sort of more intensive bit of study that we did about 3 or 4 years ago, was to take patients and really think about understanding their dynamics, really getting into thinking about can we use this mood data, this scoring data, to make inferences further down this hierarchy as we move into from sort of like cognitive activity down to sort of neuroactivity. So there's a bunch of patients here. We've got the QIDS score, and we've got basically the black line is the individual patient QIDS score over these 28-day periods, essentially. So they do that using the internet or their smartphones. They get prompted by the clinic to sort of respond to that, and we can get some pretty good information. So mathematically, it's pretty cool. Statistically, it's pretty cool. So as you see, patients are idiosyncratic individuals. Sometimes they respond, sometimes they don't. Even by prompting by the clinic, they're still missing values. So we've really got to think really hard about that data, about how we approach that data. It's non-normal data. It's bounded in the terms of the scoring. So that means that presents some interesting statistical challenges to us to think about that. But what we were interested in doing is actually taking some mathematical descriptions, and I'll show you what they are shortly, and being able to sort of characterize these sorts of time series. So the mathematical descriptions are the goodness of fit, if you like, are the purple dots, and the grey band is sort of like the goodness of fit of our models to the data. Yeah.
When the values are this QIDS score are high, does that mean that patients are happy, or?
That means they're more. So the higher QIDS score, the more risk you are of a bipolar episode. Sorry. Yeah. So the lower your score, the better, essentially. And so what we've done is that we thought about this for a long time. We took our descriptive stuff and we've taken a different. So we took a different approach. We really, as I said, we want to take this. We're thinking about approximations, thinking about what goes off in somebody's head. Can we actually characterize, at least initially, a sort of set of neuron dynamics? I'll do that phenomenologically rather than mechanistically, and scale that up to thinking about mood. So I take a total derivative approach. I have basically the change in mood through time, and then I can separate that out into a bunch of different things. I can separate that into baseline mood, the partial change in mood, something that's not independent of these things that on the rest of this, which we've got to call oscillators. So I've got baseline mood in the system, and then I've got basically two fluctuations, two systems, two coupled or uncoupled systems that may drive changes in mood that are essentially due to the sort of bipolar disorder. And what we want to do is that we want to challenge the data with this sort of framework. We want to ask the questions, is it just baseline mood? Is there actually nothing, are you just a fool to think like this, Mike, that actually, that these sorts of things are really important? But actually, is it that we need coupled? Does it that these two things, these oscillators, need to be coupled in some way? How strong do they need to be coupled? Do I need one to be different than the other? Are they noisy? All those sorts of problems. So we can think about the mathematics of this. And the mathematics of this look like this. So I've got a Van der Pol oscillator, which is just some way of describing the system going up and down, a bit like a predator-prey system, but a little bit more generic. So I've got some sort of, a little x and a little y are just the ODE version of a Van der Pol oscillator, and I've got two parameters in this that I want to sort of like try and that control whether that Van der Pol oscillator is stable or cycling, and the frequency of those cycles essentially. So, and then we want to think about, so the idea is that it's like thinking about, we've got now these oscillatory systems governing baseline mood. So we're moving from something that's going up and down in a neuron center to actually affecting cognitive mood, essentially. And we can think about that, and we can couple it with some sort of Wiener process. That's a DW bit. It's a noisy term that we're just going to kick the system at each point in time, and that could be dependent on a bunch of things. And we can couple these things together with some logistic functions, so we can make it really quite, again, I use that word, complicated. We're actually thinking about, do these sort of centers talk to each other in some way? Are they driving the system? And it turns out that when we do this fit across these patients, when we do this fit across these patients, that actually it's reasonably robust. We can actually see that the fact is that we need two oscillatory centers to actually describe the dynamics in all of the patients. Very few of them are actually always unstable. There's some, these I show you are, but there can be combinations of those oscillators where one of them might be stable and the other one might be unstable. But they tend to be uncoupled. They don't tend to have a very strong coupling. So we've got the numbers, the sort of actual data on the left-hand side. We've got what the oscillators look like from the statistical parameterization, and then the sort of predictions of that on the right hand, the goodness of fit. And we can do, we do really well at being able to capture this. And the very cool thing is that we can do this on an individual patient level, which is super cool. So some other work which we've done where we can use, rather than having the QID score as a continuous variable from 0 to 30, we can sort of group it together. We can put it into sort of groupings. And as I just said, 30 high QID scores are really bad, medium QID scores are okay, and 0 QID scores are great. So we can sort of like bin that into sort of low, 0, low, and high. And then we've got a system where we can look at the more stochastic dynamics. As you see, we've fitted generally deterministic models. We can look at the stochastic dynamics in this and really illustrate that actually we're using sort of simple Markov chains, how we can understand the system. And that's really clinically useful. So we can do this. So just going back to the very first slide I put up on this problem, is that this is a system where these patients were in this sort of like small clinical study for 2 periods, 28 days before treatment, and Emily uses imagery and cognitive behavioral therapy treatments, so they go through Emily's treatment, and then we mentored their time series afterwards. So we have this sort of comparison, and we can do that on an individual patient-by-patient level. We can look at their time series. And we can use our Markov chains to actually illustrate, this. We can use Markov chains to work out the steady states. We can work out what's the probability that you're likely to stay in a high state. What's the probability that you're in a low state pre-treatment, and how has that changed post-treatment? And that's really a really useful clinical instrument. And the great thing about Markov chains, as we get more data, we can update those probabilities as that comes along. And the next piece of work, and I really love to talk to anybody who has any thoughts about this, is actually what we really want to do is as I've described this, it's phenomenological, it's not mechanistic. We're really interested in thinking about there's lots of rich mathematics in neuroscience, and using that to be able to infer a link between these oscillators through to cognitive mood is something that that is challenging us, and really thinking about that, and not using these slow-fast dynamics, but being able to capture the sort of variance and the stochasticity in the system. Will be really cool. All right, I've nearly finished. So I'm just going to finish up about some implications. And it's a different system, and it's really cool. Another piece of work, another area of work that we're really interested in the group is thinking about biocontrol and the economics of biocontrol. So in insect, and we think about this as insect problems, as you might guess, I'm into insects. So we think about how we can use the sort of biocontrol methods for controlling pest or vector-borne disease problems. And the classic way to do that is something called the sterile insect technique. So the idea would you be mass rear your favorite pest insect, you'd blast all of that gamma and X-ray radiation at them, you'd make them sterile, those males sterile, you throw them out into the environment when they mate. Because of insect mating behaviors, they mate with wild females, they don't make any babies, essentially. So actually the idea is the population would decline. There's a modern twist on that. The idea is that you can use genetics to do that. You can engineer genetics into the mosquitoes or your favorite agricultural pest, diamondback moth or whatever it might be, which basically switches the gene on at a particular point in the life cycle and the insect dies. And normally the idea with particularly mosquitoes, which is a big there's a big area of research on this, actually, to modify male mosquitoes with these lethal genes, switches them on in the late larval stage or early pupal stage, so they die before they emerge as adults and go off and bite people, essentially. So it's the same sort of thing. The idea is to suppress the population dynamics. And there's a lot of work, and we've done a lot of work thinking about the maths about that. But one of the things that we're really excited about is actually the implications of scaling beyond the biocontrol and really thinking about the economics that play into this. So we can think of it, this is just a sort of schematic of the sorts of things that we think about. It doesn't hold any detail. So we've got sort of like, on the top, we've got dV by dt, which is the vector dynamics, and some vector ecology to do with the way in which this thing in brackets is the bit where the SIT or the lethality plays out. So we're really thinking about, well, I is one of our control parameters. How many of these modified bugs should we put into the environment to suppress the growth rate of this vector or this agricultural pest? And then there's some biology, some ecology, some demography on the right-hand side of that. And then we've got a very simple disease model, because what we want to do, we think about this as mosquitoes, it could be crop yield, but we're thinking about it as disease. A very simple Ross-McDonald framework for linking proportion of infected people, proportion of infected vectors. So that's the dynamic, and that's well worked. There's lots of huge, and you had a day yesterday all about epidemiology, and that's all very cool. And mathematical biology is rich in that area. We want to sort of like take that one step further and link that to some economics. So this is a cost function, or some functional that we want to sort of use the dynamics to minimize this function. And we've got some things in this. We've got a discounting term. So how do we value now versus the future? Do we value sick people? Do we value sick kids more now than we would our sick grandkids or great-grandkids? So we need to be able to account for discounting. And then we've got some cost things. And very simply in this, I've got the cost associated with treating sick kids. And I've also got the costs of rearing and releasing these modified insects into the environment. So that can be as complicated as we like. It's just a schematic. And I'm trying to minimize this through some sort of optimal control methods, deriving some Hamiltonians and doing all the funky maths on that, where with some constraints on what these parameters. So obviously the release rate number should be greater than 0. The proportion of sick people should sit between 0 and 1. So obviously we have some constraints on that. So we can work through that mathematics. But hey, we're now almost at lunch, and we don't need to be doing this just now. So where's that other one gone? Where's my other? I'm losing my slide. Sorry. Another bit of outreach things that we do is this. Can I make this bigger? Can I go bigger? I'm driving this from here, which is very hard. There we go. Excellent. We have this game. You can find it on our website. The only trouble now is that with the advance in sort of website security, this is a Flash-based game, and everybody's scared of Flash. So I've had to nick a Flash player for this. But we're in the process of updating this. So you can play this game if you download it. It's a Flash file, and you can just play it. And I've got to drive from here. So the idea is just to illustrate all of those sorts of things I was talking about, all of that sort of Hamiltonian mathematics through a simple game, essentially. So the idea, you've read the instructions. Here's what we do. Essentially, we're going to play this game. Let's see if this works. All right, did you do the remote recapture problem? We do this with kids a lot. We have like stick mosquitoes around, I guess the Royal Swedish Academy of Sciences might not like this thing sticking to the wall, but we would do that. And they would go off and find these sort of different coloured mosquitoes, and they'd come back and tell us how many they've found. And that's really important, because one of the really cool things about this problem is that you need to understand the size of the population you're trying to control. It's an area-wide problem, so we didn't do this. So I've got to do this from here, and my microphone is coming off my ears. All right, so I need somebody to shout out a number for me. Which one of those would you like? So the idea, we've done this mark recapture, is given an estimate of the population size. Which one do you want to choose? 7. This one? Yeah. Okay. All righty. All right, let's see. All right, there's some stuff, biology, interesting stuff about SIT, the sterile insect technique. So now here comes the interesting economics and maths, essentially. So you've got to think that there are two things that we want to sort of like play out in our cost function. One was the number of mosquitoes, sterile mosquitoes that we're going to put out there. The other one is how long you're going to do your release program for, because you're interested in sort of mitigating the disease burden. So you can on the top one, I don't know why, we can go from 1 to 10. So you can put out one one sterile mosquito for every wild type, all the way up to 10. Right? And if we weren't resource limited, obviously we would pick we would go to the full right-hand side of this. But you're trying to make this as cost-effective as possible. This is like scaling from biology to economics, right? So you've really got to think. You've really got to sort of play out in your head. So you can run your program. You can run the number of mosquitoes from 1 to 10. So somebody give me a number. 7. All right? Can I drive? Can I drive? Yes, I can. And the other one is that you can run your program, so you can see the dollar figure at the bottom is increasing, essentially. So we can run our program from 2, 5, or 10 years. Somebody give me a number? Somebody here? Remember, you're trying to do this in the most cost-effective way. 5. Thank you, Toby. So we're going to do this for 5 years. All right? There we go. All right, so that's what it's going to cost you. A release program is going to cost you just about $1.75 million, essentially. Another biological problem raises its head, and this is, we think about this as actually we've got 2 graphs now. One is the number of mosquitoes, the number of sick people. The black line is what we do in the absence of control. I think you'll get a green line in the presence of control. And the red line is the number of sick people without control, and the blue line is when we do this release. So the thing you have to think about with mosquitoes is that they're seasonal. They like it wet, they don't like it dry. They might like it warm, they might like it cold. So those black dots up there just say, am I going to prime my system to deal with it this season, or am I going to prime it ready for next season? So it's just like choosing one of those. To be honest, it doesn't make a bit of difference, but I've lost my mouse. But I'm just going to choose one so we can get through this. Right. Dynamics play out. Alrighty. So you do this control. We've got this 7-to-1 release ratio. That's quite extreme. All right. So what we see is that when we do that, we see the suppression of the population. The mosquitoes fall away, and then we stop. We run out of cash. We only ran our program for 5 years. So the mosquitoes rebound. They spread back in. They overshoot. There's lots of stuff, and they go back to their pre-control levels. All right. That's pretty nice. Maybe, if you like mosquitoes. Most people don't. Here's the number of sick people. This is the red line, is what happens in the absence of control. With the control, we can sort of like, the disease fades out. And that's not rocket science either. We know that from lots of epidemiological stuff, that if you push the mosquitoes below a threshold, that disease will fade out. What happens is that when we stop control here, it takes another 2 or 3 years before the disease actually rebounds to pre-control level. So that's pretty interesting. It gives us some indication that actually there's a lag in the system, in the disease system. But the most exciting thing is how good you were at controlling your disease. How cost-effective were you? So that's just this. How many costs, and if you were clever enough at the start, you'll notice that I had 150,000 people. I've prevented this many infections, so obviously people are becoming reinfected. But this is what it costs you to control that disease per person. $3.42. Do you think that's good or bad? Great. You think it's good? All right, so I had a 7-year-old girl do this, at one of these little outreach things we did. She sat there for about 20 minutes, and she got down to 229. I've never got down to 229 in this game. I have no idea what she did, but she worked out actually there's a critical combination. There's actually a critical combination of release ratio and times, which is about 4 or 5 and 5 years. But actually, because it's a game, we threw some little bit of stochasticity into the system, just for fun. And it throws out similar numbers, but never we got down below about 230. So if anybody does, I want to know. So that's the game. That's what you're trying to do. You're trying to make this most cost-effective as possible. But it also illustrates some of the very cool things that we can think about in controlling infectious disease. One of them is not well illustrated here because we had such a high release ratio. As I said, what will happen is that we can push the mosquitoes down a little bit. So maybe you do a 4-to-1 release ratio, something that's less onerous in terms of eliminating the population. And then we sort of play out that actually the dynamics oscillate, but actually the disease still fades out. As I said, that pushing those mosquitoes below that threshold is really important because actually it means that they're still in the system, and with it being a GM thing, there's lots and lots of controversy about whether people should be putting out GM mosquitoes. But our math tells us, our very simple math tells us, yes, it's cost effective, and yes, if you do it in the right way, you can maintain an ecosystem where mosquitoes still exist. Whether you want mosquitoes in your ecosystem is a societal problem, and that's an individual area-wide problem for a conversation, but it means we can, with our understanding of our mathematics, we can think very carefully about the ecosystem and the control that we want to deliver. Essentially, right? I have just about finished. I think I have finished, but I'm not going to be able to get back now. All right. So I have talked to you about slow-fast dynamics. and how good and bad they can be if we lose the sort of variation in the system. The idea of functional responses is critical. Again, thinking about how we move from something that happens on one temporal scale to another involves us being able to have mathematical formulations for this, essentially. And the very cool thing is that how we sort of scale between things that really do operate on different time scales. So mood or cognitive processes operating on maybe a much slower time scale than our neurofiring patterns. And moving out beyond sort of like biology into thinking about economics where we can scale in different ways, and using all this very cool maths that underlies optimal control is very useful. Thank you very much, and I'm more than happy to take questions.
Thanks a lot, Michael. There was a lot of stuff going on at the same time here. So we got another Michael in our team here that's also interested in malaria. Maybe you have a question, Michael, or... Oh, we can save that for later if you want. Any other questions? Yes, I'll give you a mic. This one. Tom Britton. Yeah.
So it seemed like you wanted to minimize that number 3.42, but don't you want to keep that number low and also prevent a lot of cases? So you shouldn't steer yourself blind on that number.
That's a really good question, right? So it really depends on what your goals are, right? So it really depends on whether you're trying to minimize the number of sick people or whether you're trying to do something that's economically sensible. So yeah, you want to do both, and you can rerun this and you can see actually how it prevents the number of sick people. But yeah, it's a really important point that actually when we think about this, what we put into our into those objective functions is really, really important. And that we think about this in terms of a dollar figure, because that's the way the world works and the way that you want to sort of convey that actually these programs are cost effective. So we do a lot of work thinking about dengue infections and thinking about combining these different control interventions. So how you could do vector control and vaccinations for dengue, which is spread by Aedes aegypti, which is an invasive mosquito around the world, and thinking about how you do that sort of how you think about that sort of control is really, really important. So obviously, dengue comes in 4 or 5, depending on who you believe, different flavors and strains, and it has some really interesting and complicated epidemiology. So the vaccines have to be efficacious against all of those strains, otherwise you cause some serious problems in populations. So thinking about how you combine these sorts of control methods together is really, really critical. And it isn't just, so it's making sure that the vector control is as cost effective as the vaccine, and then thinking about actually combining these things is as cost effective. And obviously doing something is better than doing nothing, always. But it's actually minimizing those number of sick people as well within an appropriate economic framework, and an economic constraint is pretty critical. Because unfortunately, we don't live in a resource-rich world, essentially. Thank you. That's a great question. Hey.
Yes, one last question.
So this game has an educational purpose.
Yes.
Are you showing the people some sort of glimpse or cartoon of some easy mathematics, or not at all?
The math behind this is really straightforward, actually. The piece of work was published about 5, 7, 8 years ago, where we just did, it's based on the dengue problem I was just talking about. So we did some very simple mathematics. It's just simple bookkeeping in that sense. So it is just it is an illustration of, as a game, and it illustrates lots of very cool epidemiological, ecological and economic problems. And the maths behind it, it depends on what audience I'm talking to, about how rich and detailed we can get into some of the maths on that. So I didn't want to get into, if anybody wants to talk to me about optimal control, I'm more than happy to bore you to death over lunch about it. Yeah.
Thanks a lot, and thank you, Michael, again, for this wonderful talk.
Thank you very much.